Hi everyone, my name is Fan. I'm a postdoc researcher at Aalborg University, Denmark. And I achieved my PhD study about 1 year ago, also in Aalborg University, Denmark. So the PhD was about microplastics in urban stormwater. So during the postdoc, I continued this microplastic research, but more focused on the method improvement and development. So it's a great honor to be here to share with you guys with knowledges and experiences. Yes. So today the talk was about the limits of quantification, and I do hope this will be a provocative discussion on our ability to quantify small microplastics in very complex environmental matrices. Yes, so the year 2020, it's special, not only because of coronavirus, it's also because it's a year that the global human-made products overweight all the living biomass. It was actually newly published in Nature. So among these human-made products, specifically plastic, it weighs almost twice as all animals, both from land and marine environment. So if we look at these production data of microplastics, it is estimated that by 2050, the production will reach over 30 billion tons, and which may not be a problem if the waste management is okay. But in reality, among all the anthropogenic wastes, plastic overweight more than half of the overall waste from the other materials, right? So it's not surprising people start to look at microplastics, and it's nice but not nice to hear that they found microplastics or plastics everywhere. They found it here, they found it there. And they found it in remote mountain, they found it in North Pole, in the snow, in the air, in your laundry water, everywhere. But as a researcher working in the lab, when I look at these data, the first question I would ask to the authors or to myself is, how much can I trust your data? When you report these data, do you mean that you found them all? Not only me, but also this group of people sometimes can be unkind and say that the approach you get that results might be just a cherry picking, right? You can only say you found some of the particles, but there's no proof that you found them all. While this approach might be not that problematic if the particles are large enough where you can easily see them, or there's not that much. But it becomes tricky and problematic when the particles goes really small and there are too many. I just see what I have in the, okay. There's no report of technical problems, so good, let's continue. Yes, so this is just to borrow what my colleague or supervisor, Professor Jes Follesen, he said, what they had a saying in Denmark that sometimes even a blind hand can find a grain, right? The fact that you find one or some smaller particles does not prove that you have found them all. Okay, so before going more into that very conceptual discussion, I would like to introduce you more about what we have now about microplastic analysis. So the general approach is you go out and you do sampling, right? And then you took the samples back and you do a lot of treatment, whether it's chemical, biological, physical treatment, aimed to isolate or extract the small particles from huge amount of environmental samples, right? And then you use different techniques or methods to have a closer look at the particles extracted and to see what type of plastic it is. Are they really microplastics? And then you just sitting there and look at the quality of your data and planning how should you interpret these results. So let's start with sampling. Depending on which sample you want to take, which type of sample we take, it varies a lot with the methods, right? So if we say you want to take some liquid samples, for instance, freshwater, seawater, or wastewater, it doesn't really matter it's effluent or influent. Sorry. Yes, and then you just oops. So in the very beginning, and the most common approach is bulk sampling, which means you take a container, you take a glass bottle, you go out, and then you fill the bottle with your samples, right? But later on, we think this is far away from enough, because if you say you tried your best to take 20 liters of samples from for example, a river, right? Then how can you say this represents what is really out there in that lake? So we start to build our own devices. The first version of our device, it's called volume reduced sampling, which means you reduce the volume of your sample on site, which instead of going a few liters, it can go up to 500 liter or 1 cubic meter of water within 2 or 3 hours. And then we think this is not fast enough. So we modified our device into the current version. It's so-called UFO, which means you pre-filter the water, and then it pass through a pre-screening filter, and then pass through again to the smaller pore-sized filters in parallel. But both these devices are mostly suitable for surface water or subsurface water. Then we think it might be fun to look at what's in there in the deep water. So we modified again with the units. It's so-called EU-Kraken. It can take samples from deep sea waters. Of course, these are self-made equipment. It's not everyone can at least for now they have the can see or can use it. But of course, you can also go more traditional way, like nets, or a very easily modified filter system. But this is for more cleaning matrices like drinking water. So for solid samples like sediments, soil, sand, sludge, or something, we have other equipment. For example, the first raw is sediment grab or sediment core. So they can both used in marine environment or freshwater environment. But the difference is the grab can basically already disturb your samples while you do sampling. But use a sediment core, you can build a vertical depth profile of your samples. But also equipment like shovels or also used for soil samples. Of course, human exposed into microplastic associated pollution in indoor air was also increasing of attention. So we start to sample the air samples where we use a mannequin to mimic real human breathing, or simply you just use this filter pumping system. Yes. So actually, it's not difficult or complicated to choose which equipment to use, but the problems are more about how to do the sampling. Let's take a simple example. So you go out there and you took back 5 kilos of sediment, but because the limitation of this and that, you could only analyze 200 grams from this 5 kilo. So after a few months, you got some results. But then can you really say the results is what really is there from the 5 kilo? Or is this 5 kilo really represent what is out there in the environment? Another factor is for soil samples, for example, is how deep you take the sample. If you, for example, take 5 centimeter topsoil compared to a 10 centimeter soils, you are more likely to find less smaller sized particles because by nature, smaller particles, they have higher potential to filtrate into deeper parts, right? So if you only take the top parts, it's no surprising you find less smaller particles. Then which results you should use to most closely to represent the real concentration there. So when you do the sampling, it's already setting a tune for your project. You really need to consider how representative the sample is. Let's say we are happy about the samples. After we take it back, we want to isolate out the microplastic particles. Of course, depends on the type of your samples. They should receive different types of treatment, pre-treatment. But in general, you can say they received some physical treatment, which is to isolate the microplastics with more heavier materials like minerals. So this separation use a high density solutions, which you can choose with yourself. And then use some chemical treatment or biological treatment to further clean up your matrices. And then in the end, you concentrate the samples, concentrate the extracted particles into different devices. So to put the words into pictures, it looks like this. You're happy with your samples, you take them back, you treat them nicely, gently, and then you hope you would see some clean results like this, right? The sample looks quite clean in this case. But we also have problems with this extraction, even though it sounds very straightforward and easy. Let's take a typical example, which is also the critical step in the whole processing, is density separation. We have different solutions which you can choose, right? Of course, sodium chloride, it's cheap, it's environment friendly, it's basically salt water. And these are two very good reasons of why you choose it, right? But when you choose such density, I mean 1.2 such density, you probably will get, and hopefully you will get most of the light density particles out, like PP, PE, PS, where the density is mostly below 1.1. But then how about the high density polymers, like PVC and Teflon's PTFE? And this is, you can't really ignore this part, especially when you take samples from sediments where the high density polymers are more likely to settle, right? And besides this physical density separation, another tricky issue is the process we applied on this extraction. They are not gentle, or maybe they're not gentle at all. So even if I started with one particle, and after this treatment step by steps, they are very likely to break down into smaller pieces, right? So in the end, you probably say you found 100 particles in your sample, but are they really the 100 particles in the real environment, or they are just the fragments from the one particle? Then it becomes tricky which number to put in the results. Then I believe that now a lot of you would say maybe recovery would help. Of course, it definitely will help, but it brings also other questions. For example, what do you want to use for the spiking? I see a lot of studies, they use microbeads where it's a perfectly round, small particles. So they spike that in the samples and then do exactly the same treatment steps and see how much they can recover. But these perfectly shaped small beads you can seldom find in the environment. And they're so different in shapes and so on. And they of course, they would behave very differently through all the process, right? But even if you do homemade particles, like you grinding the plastic bottles and so on, even that, they are seldom aged or weathered compared to the natural particles. So all of these saying is that the recovery they get, how much they can present the real recovery for the natural particles. Another aspect is, where do you want to spike? We often see studies, they spike particles, whether it's natural or homemade or commercial particles, into a clean sand or muffled glass beads or something, which is very far from the natural matrices. The most obvious point is you already escaped this step of organic matter removing process, which for a real sample, it's really the main goal to remove it. So the whole process, can they really comparable? And also, a lot of times the homemade mattresses, they have different texture compared to the natural ones. So it's really about representation. Again, let's assume we are now happy with the extraction method, and now we need to identify the extracted particles. So to choose which technique to use, normally we choose that based on the particle size you want to look at. If the particle, they are big enough where you can just use something to manually sort it out, then optical microscopy is, of course, a good option. But when the particle size goes down, this method, it's reaching its limitation, where imaging, whether it's micro FTIR imaging or micro Raman imaging come to play. So a third option is a thermodegradation method, paralysis GCMS. So it's mainly these 4 methods on the market now. But if we look at the data reported so far, the small particles mostly found in the environments, they are small size, between 10 to 300 micro. So that tells a message that the optical microscope really has its limits on microplastic analysis. If we look closer at these analytical techniques, optical microscope, although it's missing a lot of small particles, but it's fast, it's cheap, but it's very operator dependent. If this particle seems to me, for example, it is a polymer, it's a plastic, but you may not think so. So you would just discard it. So it's a potential source for the biases. But micro FTIR, it can go down to a few micro in size, and it provides morphological information, particle number information, but it's expensive and it's also time consuming. Paralysis GCMS compared to micro FTIR imaging, it can give you more accurate measurement in mass concentration, but it has no particle info, number info, no morphological info. So basically means once you shoot a sample in this machine, they are absolutely gone. There's no way back. So be careful when you shoot your samples into this machine. Probably you want to get more morphological information before you shoot it, right? So for myself, I work mostly with micro FTIR imaging. So I will go further down with this technique. Here you can see a window of a diameter of 10 millimeter. So you scan that under micro FTIR and you get IR imaging, right? So among this sparkling, shiny IR map, how do I know which one is plastic, which one is not? About 3-4 years ago, our approach is very manual. So what we do is we, so we zoom in here, like very little zoom in, zoom in, zoom in. And then you found one particle very suspicious for me because it's red or just my gut feeling. And then you zoom in also the IR map. And then you click there, click around, and then you get an IR spectra. And then you compare this IR spectra with the reference spectra from the library. Do they look similar? Do they look exactly the same, or do they look very close? If so, I say, okay, it's a plastic. Then I note down on my small notebook. And then I go back again to the visual and then do the same job, find the next suspicious particle and check the IR spectra and then do the comparison again. So you guess how much time it would take to finish analyzing this whole piece of window? It's approximately 1 week. So we were crying for automation. And our dream came true. We made our own software and now it's integrated with a AWI approach. You can also find it online here. So the idea of this software is it takes the spectra from every single pixel from this image and it compared that with the reference spectra and give you a score. So if the score above the threshold you set, then it will be listed in the microplastic table. And this software did an amazing job because this 10 millimeter window has a pixel resolution of 5.5 micrometer. That means it has over 3 million spectra to compare, right? So there's no way, manually you can do this within four hours. So we are very happy about that. But the question becomes, does automation solve all the problem? Unfortunately, the answer is no. Because if we say with the same sample, the manual sorting or the manual analysis gives me 100 microplastics. And with a software analysis, which contains 100 reference spectra, I will get 500 microplastic particles, which is good and fine. But if you enrich the library spectra from 100 to 300, it will give you probably thousands microplastics. This is a huge variance, not only in absolutely particle number, but it will interfere a lot with the polymer composition. So I hope now you get a better feeling of what I mean, that the approach is more or less like cherry picking, and why the fact that you said you find one or more small particles does not proving that you found them all. But if we can't find them all, so then what is really our limit of quantification? Different people, they have very different opinions, but definitely it's not the smallest size of the filter or the smallest particle we found. And to put this in a broader picture, we know that microplastics research, it's a multidisciplinary research. And among this whole picture, why analytical method is important? It's because when this extremely increasing public concern is making the politicians of making any regulations and so on, but to reach the level of giving out any regulations, they request more data, because the current data, they are still very fragmented. They request more data on the health or safety studies, whether it's on to the individuals or the environment. So that requires the risk assessment from the toxicity research and so on. But in order for them to do a realistic study, they need input from us to know what is really there, right? Maybe 2 or 3 years ago, people would say that analyzing microplastic is still infant stage. Maybe we are closing to toddler now, but absolutely not adults. So the analytical methods still need a lot of improvement in order to send the reliable message to do this safety assessment. So my advice to general, also this is borrowed from my supervisor, is let's be honest and document what you did, because nobody can really now do quite right. And thank you.